All right, this morning we're going to talk about calcium. I'm going to try not to get off on any tangents and see if I can finish this whole thing in an hour to an hour and a half. Um, there's so much to cover, and I want to make sure that everybody has more of a complete picture because a lot of this is completely overlooked um, in others' discussions on calcium, while some of it's covered real well. Um, so let's dig right into it. I got some notes here so I can try not to forget anything. Um, with as far as calcium needs go, we have the soil needs, we have the needs of the plant or the crop, and we have microbiological needs. Um, as far as environmental factors with calcium, um, water is a huge one. Calcium's a large, heavy molecule and it moves downward. Um, when you get a lot of water, if that calcium is not tied up by microbes, um, it tends to get washed downward with gravity. So um, you have those calcium needs, and um, those calcium needs in the plant and the microbes are far more extensive than what I've heard anybody discuss. And we're going to get into that after we cover the soil. Um, so for soil, um, we can go back to William Albrecht's work, and that's probably the most reliable um, calcium work as far as building a base soil and making sure that you have a sufficient amount in that soil. Um, and so basically, what his work um, was saying is you need to have a minimum amount of calcium in your base exchange, and with that being 68 in his work. Um, I have found that a little past 70 is actually usually better, depending on your soil type and what's going on. But when you get a little bit more in there, there's a little bit more available for the microbes and the plants um, for the stores that they need to take up. And we're going to talk about that later in the discussion. But basically, his work was stating you need to have 68 percent calcium in the base exchange, 10 to 13% magnesium, and 5 to 8% potassium, with the remainder being taken up by hydrogen and um, cation metals, micronutrient metals. And like I said, calcium being the mother of all those and the kind of the controller of the soil structure, it does actually help to have a little bit more than what Albrecht's minimum recommends, and that is minimum 72 to 75%, if you have a rich microbial life, and vigorous plants tends to be a little bit better. Now, as far as contained medias, such as rock wool, cocoa, peat, um, living soil, etc., you're going to want to um, shoot for those same numbers I was talking about, the 72 to 75%. Um, those medias tend to have a very extensive cation exchange capacity, and loading that up with calcium helps prevent compaction, and it also gives a little bit more availability for the things we're going to talk about later, which are extremely important. Let me check my notes here. In heavy clay soils, it's especially important to have your calcium saturation where it needs to be on the base exchange. Clay colloids tend to lay flat like that on each other, and these faces are negatively charged, and the ends tend to be positively charged. And they'll sit like this, overlapping each other like that. And when calcium comes in, it opens it up and stands them up like that. So then your colloids are stacked like I-beams. And the minerals are able to move in and out of those ionic spaces and are far more available to the microbes in the plants. And it also opens it up for air so that the clay can breathe. When you get too much magnesium in there, it collapses those and lays them flat and tight and causes a lot of compaction. So if you're having issues with compaction or water penetration, get your soil tested and see if you don't have too much magnesium there. That can be offset by simply adding calcium. The calcium will push the magnesium out of those clay colloids, and at the same time it'll free up nitrogen because the magnesium is going to be holding on to nitrogen in there. So for different soil types and different soil issues, there's different calcium supplements you can use to correct those. For heavy aluminum acidic soils, wollastonite, which is a calcium silicate, can help those a ton, because what it does is that silicate will then bind to the aluminum and tie it up. Your pH will come up a little bit as the aluminum's not active in the soil anymore. And then you have the calcium available for the base exchange while the silicate ties the aluminum up. For higher pH soils, calcium sulfate works well. Calcium sulfate plus protons, so organoacids, will help acidify higher pH soils and will help open up clay soils. If your clay is high pH, the calcium can't get in there anyway, and using organoacids, specifically calcium chelated calcium organoacids, will get into those clay particles, open them up, and allow calcium to do its work, to flocculate those clays. For acidic soils that aren't as high as in aluminum, calcium lime's an excellent one. And for high organic matter soils, calcium lime's an excellent one. The carbonate in high organic soils, the carbonate can end up being an excellent anion in there to help with microbial life. Sorry, my phone shut off that I have the notes on. Okay, let's see. Okay, so calcium, as we talked about in how it opens up clay particles and whatnot, when it's in sufficient percentage in the base exchange and the cation exchange, your water holding capacity is significantly higher. because of the way that calcium regulates the hydrophobism in the soil and the aggregate particles. All right, let's see. So let's talk about time to availability on the calcium amendments. All your rock powders, your crushed and micronized, your availability is going to be dependent on the micronization. As some highly micronized products are immediately available to some degree. Now, when you're talking about amending soils with crushed rock powders or micronized rock powders, amending the soil rather than suspending them in solution and watering them as a solution, making them more highly available immediately. When you're putting them in the soil, there's a 2-week to 90-day curve where they're available depending on how micronized or crushed they are. But if you want your spring garden beds to have sufficient calcium when you first put your plants in, you need to be amending those beds a minimum of 30 days prior to putting those plants in. And then it's a good idea to amend every 30 days throughout the growing season, just small amounts. And simply because, like we talked about earlier, calcium sinks. And the more rainfall and the more water your garden has, and the less microbially what's the word I'm looking for? It's a little bit earlier. Calcium that the microbes haven't already taken up is going to fall through the soil structure. A lot of organic matter and high cation exchange capacity slows that down, but it's still going to fall through the soil. So regardless if your soil test at the end of last year or the beginning of the year said you had enough calcium, by 3 months later, you may or may not, depending on what's going on in that soil. So be aware of that. It is always falling. So, there's a high need at the surface, at the top 4 inches, for calcium, and that needs to be maintained, or things start to suffer. Okay, so let's get to the needs of the plant. So we all know structurally that calcium is extremely important. We know that it's part of every cell wall, that it provides the structure and strength of the cell walls. And it's also a big part of the plant's pectin. Calcium is what makes the pectin thicker, to where pests and pathogens can't get through it. It provides a barrier in addition to the plant wall barriers. Calcium is a regulator of plant growth. It's extremely important in the transport of other nutrients, in architecture and structure of the plant cell and the cell walls and the plant itself, especially the roots. And calcium's required at the root tips without it, there is no cytokinin production, and root growth comes to a stop. It's required for root hair growth. It's required for lateral root growth, etc., etc. Now, let's get into what's not covered as much that I've heard as far as the plant requirements of calcium. So, every single cell of the plant has extensive calcium stores. And if calcium is limited, the plant will fill the different requirements at different rates, and as it becomes more available, then it'll allow more to go to different places. But it's going to prioritize where that calcium goes if it's in limited supply. Now, it's a structural part of the cell wall. It's integral in the stability and the stability of the cell walls. There's multiple types of intracellular, intercellular calcium storage. Let's say vacuoles, okay? As calcium becomes sufficiently available, these calcium stores are increased and filled up. These are within the cell cytoplasm. They're within the cell walls as well, these additional stores. And they're also within the nucleus. These storage vesicles are full of an acidified, they're an acidified calcium storage. A lot of times it's phosphate in there with it, and they're in the low to mid 5 pH range inside it, so it's completely soluble and easily transported in and out of the vacuoles and vesicles. These are within and without the cell and within the nucleus. They're everywhere in the plant, and when calcium's sufficient, they're saturated within the plant. These are used for signaling, for controlling gene expression. They're used during pathogen attacks. They're used during pest attacks. And when these stores are not sufficiently saturated within the plant and the plant cells, then the plant, even though it may have sufficient calcium in the cell walls and in the pectin and whatnot, say, an herbivore attack comes in and starts taking chunks out, like a grasshopper. If the plant has sufficient calcium, it's able to heal those up quickly. There's no issues. You see that around those, it'll turn brown. Around something like that, it'll turn brown quickly and stop any further entry through that open wound. It closes it quickly. That's all dependent on calcium. Plants have a nervous system. It's calcium-gated glutamate channels, just like we have in our brain. There's different types of receptors in there, but calcium's the signaling molecule. And when a lot of calcium can be used up very quickly, and the ions move to different places in the plant as it's shuttled in microseconds to a different part of the plant through um, gated ion channels. Anyway, so if there's not a saturation of those vesicles and vacuoles throughout the plant, the response to these things is nullified quickly. Now, like I said, it's also used for gene expression. You have calcineurin, calmodulin, calcium-dependent MAP kinases, calcium-dependent AMP kinases, and those all control expression of genes. They control epigenetic expression, and they control expression. They control the expression of what DNA is expressed to according to what environmental stimulus. Without these extensive stores within the plant that are saturated with plenty of calcium, this epigenetic attenuation to stressors is limited. You know, when the calcium just gets pumped into the plant by the microbial associations at the root because there's sufficient in the soil, it's sufficient in the microbes, and then the plant has sufficiency for its growth, for its neuroregulation, for its signaling, and now for its gene expression, and epigenetic expression, and pathogen and pest resistance, because it has just these boatloads of calcium to respond to any type of stress, abiotic or biotic. It changes the plant entirely, and you get a plant that's operating at an 80+% photosynthetic rate, producing tons of secondary metabolites. Regardless of what plant it is, it becomes a medicine when that's the case. Okay, so you kind of get an idea of how important that is beyond any type of structure or architecture in the plant. It's so much more important beyond that, but you can't get there until you have the structure and the architecture. The plant will obviously allow some to go to those because it has to have a minimum amount of those, but it's going to take it and put it to structure and architecture first, predominantly, while allowing a little to go towards those vesicles and vacuoles for those purposes we talked about. You don't see a deficiency in the plant if the architectural and structural and that minimal is there in those vesicles and vacuoles in the storage. You don't see a deficiency in the plant. But the plant is not performing where it could be, right? If you start checking your bricks, you'll see a deficiency there because you won't see that plant go past a 6 or an 8, okay? Once you get those extra calcium stores filled and more created and saturated throughout the plant, you'll see the bricks respond. You'll see the bricks go to 12, 14, 16, 18. You know, when you're getting your plants up at that 16 and 18, it's producing all the secondary metabolites that it needs, all that it wants. That plant has aromas, colors, and flavors that you won't see until it gets up there. And it won't get there until those internals are full and saturated. All right. To get there, to get those full and saturated, you need a heavy microbial association at the roots. Fungal, bacterial, the works. Okay. And those need to be fully saturated. We're going to talk about those next. Those can't get fully saturated until the soil is completely taken care of first. And then the plant basics are taken care of, like we were talking about, the structural, the architectural, and the minimal of the other. Okay, once that happens, okay, then the microbes can begin filling their stores. The microbes can't really begin filling their stores until that plant gets to that bare minimum. It's a tripartite association, and I'm going to tell you why. Because the plants, in order to increase the density of their root associations, they will take calcium that they have and display it at their roots to attract the microbes. The microbes associate, the crosstalk begins, and if the plant has enough bricks, if the plant is satisfied at that bare minimum and has the 6 to 8 bricks to start feeding the microbes can begin assimilating more calcium in the soil. There has to be this feed and this crosstalk for that to be amplified. Okay. And getting your bricks up in your plant is the best way to do that. There's biostimulants that will greatly speed that process. Microgasm's one of them. That increases that. It will increase those root associations immediately. Now, what I've seen is that with that product alone, unless you have absolutely perfect micronutrient availability and whatnot, it will take you past the pathogen resistance point, but it can't really on its own get you past the 6 to 8 bricks unless your mineral nutrition's pretty perfect. What I have found is that, if your soil to begin with is lacking that mineral nutrition, you can get that microbial life going, get those associations going, and foliar feed some micronutrients at the leaf level and a little calcium. And what that will do is it will amplify the signals that the plant's giving to the microbes, and the microbes then work overtime. It increases that bricks even temporarily for 3 to 5 days. Those microbes start working overtime and they start assimilating everything out of the soil. They start super loading their structures, which then ends up getting the plant super loaded. The plant won't get super loaded until the microbes do get super loaded. Okay, there's that minimal feeds the minimal, and then stimulating that, the microbes will superload, and then the plant will get superloaded, right? And that process can happen extremely quickly inside of 24 to 48 hours. If you have the calcium and the micronutrients available to the plant and the soil, right? And there's many ways to do that. If the soil's being stubborn and you're just getting, these associations going, foliar is an excellent way to attack that. And what it does is that signal you give through the foliar is amplified through the soil. What will happen is those little tiny bit of nutrients that stimulate the plant feeds those microbes, and they go crazy and go get everything that plant that needs out of the soil and brings it right back. Superloads themselves, superloads the plant. So let's get to those same structures down in the microbes, okay? Your microbes, same thing. They have a structural and an architectural need for calcium. Beyond that, though, your microbes, your fungus, and your bacteria both, they still have those same calcium requirements for signaling. They still have your calcium gated ion channels, the glutamate channels. They still have the calmodulin regulation. They still have the calcineurin sensing. They still have the vesicles and vacuoles within their bodies that they need to fill up in order to be primed for all this signaling and DNA expression, epigenetic expression, and changes within the as those genes are expressed, right? So calcium is the greatest determinant of the acetylation or deacetylation of these genes, these proteins. It's the greatest determinant of the phosphorylation of these proteins, the phosphorylation of different enzymes activating or deactivating them. Calcium's controlling 80 to 90% of that in plants, microbes, and humans. Okay, in every cell type. This is what's not discussed as heavily, and I don't know, I'm not sure why. But all of these things are dependent upon calcium. Let's get into mycorrhiza to start with, okay? Because mycorrhiza, if you have them in your media, in your soil, they can be the biggest determinant of plant nutrient acquisition and plant health that there is. And there's a big reason why, but we'll go into that. So, mycorrhiza have extensive calcisome storage, acidic, lysosomal, or vacuole, or calcium storage, and it's almost always calcium phosphate in the acidified calcium phosphate in the mycorrhiza. They store these both within their hyphae and without. They create a biofilm around their hyphae for the transportation of nutrients and microbes and other fungus. That creates an environment for certain types of microbes to be able to travel and store and move nutrients in that biofilm. Now, within their hyphae, they also contain these calcisomes, acidic calcisomes, but they also harbor other fungus and microbes. Okay, most of what we see happening with mycorrhiza is not necessarily directly due to the mycorrhiza, but rather due to the consortium of biology that the mycorrhiza brings to it. If you're looking at soil that's heavily populated with mycorrhizal associated roots, and you're just looking at the soil in general, and you're not taking a sample of those roots with the mycorrhiza attached and looking at that, you're missing the entire picture. Because when mycorrhiza is involved, they're calling all those surrounding microbials to that mycorrhizal network, and then creating the perfect environment for all those other microbials to move, to acquire and store nutrients, and to access the plant roots directly, and be taken up through the visophagy process this way as well. So mycorrhiza is the logistics. They are the logistics and the framework for all these other microbes to access the roots and to bring all those nutrients and store them and have a safe environment to coexist. Like I say, the mycorrhiza are able to separate to microbes that don't get along with each other, and have certain types of microbes in that biofilm, while others are within the hyphae themselves, both other fungus and bacteria. Now, the mycorrhiza have extensive stores of the calcium phosphate, the acidified calcium phosphate in vacuoles, vesicles, lysosomes. And they will trade that off in boatloads to the plant as it needs it, and to other bacteria within the consortium, etc. And it's used within them for signaling as well. All of these same type signaling molecules and processes are within the mycorrhiza as well, and extensively used. And in this whole interplay, what the mycorrhiza are doing is they're collecting genetic data from all these other microbes. As these other microbes use the mycorrhiza as a logistical channels and freeways and whatnot, the mycorrhiza is sampling their DNA, storing it in the multitudes of nuclei that these mycorrhiza keep within their hyphae. So once those bacteria and fungus have come into the mycorrhiza and used them or their biofilm as transportation or harboring, the mycorrhiza then have their genetic data, and should they die off, they can use that genetic data can be transferred around in the gene pool to other microbes to continue the work at a high pace. So, and for all this to happen, all this interplay to happen, it's so highly dependent on calcium. And that's the third to last place. Those microbial and mycorrhizal stores is the third to last place that the calcium goes. Okay, it can't saturate those until all those ones we talked about are saturated first. Okay, and then once those microbial and mycorrhizal are saturated, then the plant gets super saturated in all of its storage vesicles and whatnot, and then it'll create more of those, and the plant health and the secondary metabolites and the bricks exponentially increase. Okay, but every one of these systems is dependent on calcium, and you stop seeing a calcium deficiency halfway through that cycle. Okay, so when you stop seeing the calcium deficiency, you still need twice that much calcium or more available to the plant. It doesn't mean that if you were at 70%, you can't get to 140%, right, in the base exchange. But what you need is you need twice as much available to the plant. So you need to get that base exchange up to 75, even 80, if you really want to push things. That's the way to do it. Let's see where we're at here. Okay, so now, because of all these other calcium needs and how much signaling and defense depends on those needs, you can see a plant that doesn't look to have a calcium deficiency at all, because that first half is taken care of. So the plant's not telling you it has a deficiency. However, its response to a pathogen or a pest is not going to be anywhere near the same as if you have that full circle saturated, right? So the need beyond the site of a deficiency is significant, and if you are seeing any type of damage on your plant, whether it's pathogen or pest, right, you don't have that full circle need satisfied, period. You can call in all the microbes that you want with the biostimulants and the sugars and everything else. You can call them all in all you want. They can suppress a pathogen that's there, but then 2 weeks later you start seeing signs of that pathogen again. The reason why is not because you didn't get the microbiome there. It's because you didn't satisfy that microbiome or the plant, right? And the microbiome has to be satisfied on that extraneous stores before the plant can be, right? If you're seeing this return 2 weeks later or a month later, when it did get suppressed, okay, the biology's there. The biology suppressed it, but the biology ran out of calcium because there wasn't that excess there. You know, it brought in everything it could. It put out these biological agents, these antibiotics and whatnot that stopped the pathogenic infection. You know, with my product, you stop the quorum signaling, so you give the biology you call in an upper hand as well, because it'll call the biology in, and it'll stop the quorum signaling along with several other things that it does all at the same time. So you just stop the infection, okay? You stop the disease. If you don't have enough calcium there, a couple weeks later it comes back. It starts to be seen again, not because you don't have the suppressive biology there, but because the quorum, right, that product's only live in the system for so long before it's metabolized. And after it's metabolized, that quorum signaling is not blocked anymore. And if those microbes don't have enough calcium to fulfill those excessive stores they need and then give to the plant the excessive stores it needs, if that's not there, then those symptoms can creep back in. Okay? So if you see that, just add calcium. You probably don't even, you may or may not even need to stop the quorum signaling again. I advise it because it's an instant stop. There's no more pathogen virulence. It doesn't go any further if you stop that quorum signaling. But add calcium at the same time. It doesn't even have to be a lot, but if you're having that issue, make sure it's a type of calcium that's going to be immediately available. Either a highly micronized powder that's dissolved in water and kept agitated as it's spreading, like a micronized calcium sulfate or a micronized calcium lime, or get an amino chelated calcium or an organochelated calcium that are completely soluble, and apply those so it's immediately available. And then if you're talking soil rather than a contained media, you could also sprinkle a crushed rock powder at that same time, because it's not going to be available for 30 days, but what you put on it soluble can hold you over for 2 weeks to 30 days as that's coming in. Once you get that calcium in that top layer, where that top 4 inches where they have access to it, the microbes have access to it in that extra sufficient amount, you'll never see the pathogen come back again, period. And here's why. Most things that we consider pathogens are not actually pathogens. Most of them are actually beneficial endophytes. Fusarium being a key one in point. Alternaria, another one. They're everywhere. They are endophytically associated with almost every plant they're found in contact with. When you test across 400 species of plants, and all but 14 of those, Fusarium's acting as an endophyte completely. Never, ever acts as a pathogen. Okay? And then you have, this small number in that group of 400 where it starts out as an endophyte, and then during some course of the plant's life, it switches to a pathogen and negatively affects the plant. That's how most pathogens work. And guess what is the primary determination in that genetic switch from symbiont to pathogen. 80% plus of that in that symbiont, could be pathogen, is controlled by calcium. If calcium ever becomes insufficient, and Brix drops below a given number, usually 6 for pathogen, 4 to 6. If you get below that number, the pathogen, so there's a ton of different chromosomes that have pathogenicity genes on them. Okay. Calcium controls 80% plus of these. Okay. If calcium becomes insufficient, the plant or the endophyte switches its genetic expression and starts acting as a necrophyte, right? And then it will release mycotoxins, kill the plant, and eat the plant matter. Okay, calcium controls a whole ton of that. Now, nitrogen and carbon acquisition and oxygen acquisition control those as well. But most of it boils down to a calcium control on those. Okay, calcium's what's controlling the phosphorylation or acetylation of a protein or enzyme, and pushing the expression of these genes in different directions. Okay, when calcium's sufficient, they don't, those controlled expressions don't happen. Okay. Now there's a few other triggers outside of that, but it's highly, highly, highly dependent on calcium. Okay, especially fusarium. Well, they all are. They all have the same metabolic framework within them, and calcium controls that heavily, just like it does in our bodies and in the plants' bodies. So stopping at the Albrecht, when you get your soil sample back, stopping where it says I have 68-70% calcium, I don't need to add any, that's not a smart idea. Okay, first of all, take a soil sample of the top 2 to 4 inches and take a soil sample of what's below that down to a foot, in different segments preferably, but look at that top 2 to 4 inches. How much calcium's there? If you don't have 70% or better there, then you're not satisfying that anyway, right? And so, because of all these other things we've discussed, that's why I recommend people to have it more up at 75, right? And then, if you have any type of expression anywhere of any pathogen, even a virulent expression, look at what's going on with your calcium. Okay, look at where your magnesium and your potassium are that can compete with that. Okay, because if your calcium can be at 70, but if your magnesium's at 15 or 18, you still need more calcium. You got to get that magnesium down and get some protons into that, after that, to displace some calcium back out if you need to, right? There's a great interplay with calcium and protons as well, so that's where pH comes in, right? If your pH is too high, calcium's just not available. It's going to be locked up. There's no protons. The protons, will exchange with the calcium to a greater degree than the other cations. Okay, let's see, where am I? I know there's a couple points I'm missing, but I was able to get through a lot of it because I didn't get on tangents. Okay. Okay, without going into great detail on everything that those calcium stores are do, it's pest pathogen resistance, as well as abiotic stresses, saline stress, drought stress, heat stress, etc., etc. Although all the sensing and the reactions of those are highly dependent on calcium. Um, if you have those stores extensively saturated throughout the plant, both intracellularly and extracellularly, um, the plant can respond to any stress, regardless. Because if those stores are sufficient, it can instantly close the stomata. It can instantly stop uptake of things. It can instantly respond to saline and osmotic stress. It can instantly respond to a pathogen invasion, at the stomata or any other part of the plant. Even, Fusarium likes to go in at the cap cell of the trichome. And if you have a sufficient calcium stores, the plant can respond to that and cause that uh, that cell to die and prevent entry beyond that point by the pathogen. Something just turned on. I'm not sure what. Anyway, okay. So takeaway here. Oh, so let's get into to plant and fruit quality is highly and plant storage time or fruit storage time after harvest is highly dependent on calcium. Also on phosphorus, highly dependent on calcium. When your calcium is sufficient for all the full circle, your vegetable storage time is incredible. When it's just full to where you don't see any deficiencies, you don't see blossom end rot or anything like that, but you don't have all the rest of that circle filled. Let's say your tomatoes are going to keep for a couple weeks tops, right? And then they're going to start getting wrinkly and soft and mushy. Okay, when you fill that thing full circle, your tomatoes keep for 6-8 weeks before they even start to get soft or any wrinkles on them, right? A huge difference. Your lettuce will keep for 5 weeks in the fridge if that's full circle. If it's just to where there's no deficiency spotted, you got a day, 2 days to a week in the fridge, and then it's getting mushy and gross, right? There's a big difference between what we see as not a deficiency and a true sufficiency. So we have to have that full circle for a true sufficiency. And when we have that, the plant suffers from nothing. The plant is operating at 80+% photosynthetic rate, and the plant is producing secondary metabolites that far exceed anything you'll see from that plant outside of those parameters, right? This is so highly dependent on calcium because you have to fill that other half of the circle, right? There's deficiency, and then there's true sufficiency. Okay, there's another whole half of the circle. At another time, I'll get deeper into each different aspect of this. But know that these pathogenic switches are so highly dependent on calcium. And that's why you can get rid of a pathogenic problem for a time and see it return. Okay. The calcium in the top 2 to 4 inches of your soil.